I'm Anthony Prieto.
I'm Brian Naboa.
I'm Charles Uhas.
And I'm Muhammad Ali, and we're trying to knock out cancer by characterizing protease activity and selectivity. Papa! So cancer is a growing problem in the world, and nowadays you can find it anywhere in the body. You can find it within your lungs, pancreas, skin, and many more areas. But what we, in our research project, are deciding to focus on is what happens when cancer metastasizes. Now, metastasis is the process in which cancer goes from a localized area and escapes that localized area and moves throughout the body. Now, cancer is, focusing on the figure first of all, we have our healthy cells, the ones on the side, that are containing the cancer cell, the middle blue cells. And then what happens is the cancer escapes, and that's the metastasis. And that's when the cancer cells start to flow throughout the body. Now, cancer itself is already a hard problem to deal with, and it's already hard to solve, like cure. And then when metastasis occurs, it's actually even worse, because you can't now just focus on one area. You have to focus on different parts of the body, and this decreases the chance of curing the cancer dramatically.
Our study of interest is in how the protease is involved with the spread of cancer. As you can see in this diagram, these are the cancer cells and these are the healthy cells. And what's connected between them are the cell adhesion substrates. Proteases are involved with cutting these cell adhesion substrates in proteins. They're also in charge of day-to-day biological processes in the human body, such as cell growth, cell migration, blood coagulation, and programmed cell death. The problem is that when there's a presence of cancer in the body, there's an increased amount of these proteases found around the tumor. And now, because they do what they do in cutting these cell adhesion substrates, they tend to be overworking now. So now, too many of these proteases are cutting the cell adhesion substrates, which gives an opportunity for these cancer cells to break out from around the tumor area and around the body. Our protease of interest is specifically human kallikrein 7, or HK7. HK7 is a very common protease found in cancer, different types of cancer. This diagram shows how HK7 is involved with the spread of cancer by first binding with the substrate, as labeled in the previous slide, and once it's bind, they cleave it, which leaves the opening for the cancer cells to spread. We're going to be studying 5 cell adhesion substrates labeled S1 through S5. S meaning substrate. We're chosen these 5 substrates because they're very commonly found in the human body. Our goal is to determine which of these 5 cell adhesion substrates that HK7 likes to cut at the highest rate.
So in order to do this, we had to use a method that would clearly tell us whether or not the substrates have been cut. The method we use is called FRET. So what we do is we take our HK7 enzyme, we combine it with our substrates, each of the 5 individual substrates, and then we have a fluorometer that excites it with an ultraviolet light at 408 nanometers for the wavelength. It uses this light because this is the light that will excite the cyan protein. If the substrate is connected, or if the substrate is cut, all the energy will stay with the cyan protein, and we will see a cyan fluorescence color. If the substrate is still connected, the energy will transfer through the substrate, and we will see a yellow fluorescence color. This is what we're interested in because it will clearly indicate whether or not the substrate has been cut. So using this, we will be able to determine which substrate gets cut the fastest. So before we did this, we had to first create our HK7 and our substrates. To create our substrates, we used a plasmid, which is a circular DNA chain that has a promoter and our substrate of choice. The plasmid was then inserted into a bacteria cell, and the bacteria cell acted as a host. During the peak growth phase of the bacteria cell, we introduced the promoter, which in this case was a simple sugar called arabinose. The arabinose prompted the bacteria cell to create the substrates. After the substrates have been created, we then had to break down the cell membrane of the bacteria cell, and then we isolated and purified our substrates, leaving us with a fluorescent yellow solution. It was fluorescent yellow because all the substrates were still connected, which is what we wanted. To create the HK7, we did almost the exact same process. We had another plasmid, however, it was a different promoter, and instead of the substrate, we had the HK7. This plasmid had to be inserted into a yeast cell. We had to use the yeast cell because bacteria cells cannot create the complicated HK7 protein. So once again, once the yeast cell was in its exponential growth phase, we added the promoter, which was a different simple sugar called galactose, and then we went about the same way, breaking down the cell wall and the cell membrane, and then we isolated and purified our HK7. This left us with the isolated substrate and the isolated HK7 that we needed to continue with the experiment.
After we isolated the substrates, we mixed in HK7 with each individual substrate and put the mixtures into a fluorometer, which measured the emissions of the fluorescent yellow and fluorescent cyan. With this data, we were able to extrapolate a percentage of how much substrate was cut in each solution. As demonstrated in our y-axis, our x-axis was each individual substrate, including a negative control, which was just to show that our machine was working properly. After 2 hours, substrates 2 and 3 were fully cleaved, and this was the time that we used to compare all other substrates amongst each other. So substrates 2 and 3 were cut the fastest by HK7, which is what we were looking for.
So now knowing this information, what we want to do is add an inhibitor. So an inhibitor is a protein that stops all functions of a protease. So as you can see in our figure, we have this Pac-Man looking figure, which is our protease, the star, which is our inhibitor. Now I want you to imagine the star gets stuck in our Pac-Man. Now Pac-Man can't eat anymore. So that's the same exact thing that happens with a protease. Once an inhibitor gets stuck in the protease and binds with it permanently, the protease cannot function at all. So we tested this, and what happened was, our S3 right here, when we added the inhibitor right here, you can see it dramatically decreased the amount the HK7 cuts the substrate. It's almost equivalent to our negative control. So now we know, but you may think this is a good thing, but it's actually not, because our HK7 has important bodily functions, and we still need some of it. We can't have it completely not cut the substrates. So to conclude, S2 and S3 are our substrates that are cut the fastest. So now what we want to do is to create an inhibitor that decreases the overactivity of the HK7. So the way we would do this is first we make an inhibitor that first of all targets only HK7, because the current day inhibitors we have cannot focus completely on just one protease. It somewhat binds to other proteases. And like as I said, So then secondly, what we need is to find a certain concentration of an inhibitor to put in that localized area where the cancer is. And the reason why I say a certain concentration, not completely wipe it out, is just like what I said previously. HK7 is still has important bodily functions in our body, and we still need it for regular, everyday things. So this is our future plan to continue off with this research. So we'd like to thank the Jack Kent Foundation for even helping to sponsor this program. We'd like to thank our advisor, Patrick Daughtry, our graduate mentor, Jennifer Guerrero. We'd like to thank all the program advisors, Maria, Jens, and Christophus. We'd like to thank our RAs, Sarah, Corey, Spencer, and Stephanie, if she's not here, and then as well as UCSB and the CNSI program. And we'd like to open up the floor to any questions.